So thank you, Thomas, for the introduction and also for inviting me to give a talk to this school. So my talk is very different from what you have seen. First, because it's optics and not all these other modalities, and also because we don't do things for the clinic. It's things for research on the brain, on how the brain is working. And so why should we use optical methods to study the brain? Well, to take advantage of the high spatial resolution, for example, of the optical method. So it's a method that gives sub-micron resolution, and you can see here on this brainbow image that you can see very well the cells and also the dendrites and axons. So optics give a very, very nice resolution, the best that you can hope for to study the brain. And also we'll see that using optogenetics, it's easy now to target specific cell types, subcellular and some regions. So it's interesting to use optics also to refine this. And also it's a non-invasive method. So compared to the other method that people are using to study the brain, which is electrophysiology, optics brings some nice advantages. So in which fields people use optics to study the brain? So one first type of application is to do morphological imaging, so to study the structural connectivity of the brain. So for example, you have examples here where you tag one neuron and you can see where is the neuron projecting in all areas of the brain. And the goal of this kind of technique is to build a structural description of the networks of elements and connection in the brain. And so the other type of applications, which I'm more familiar with, this is the domain that I work on, is to image neuronal activity. So as you know, the brain is a highly dynamic system, and the dynamics, they range from milliseconds to days. Milliseconds, which is the time at which action potentials are emitted and the neurons are connected, and days, which is the time where we learn things in the brain. And so we need to be able to look at all these dynamics. And you know that the building block of the brain is the neuron, so which is here schematized as a triangle. So basically we want to see these neurons, to image this neuron, the activity of these neurons on all these dynamic range. So this is the challenge for imaging techniques. And why do we want to do that? Well, we want to do that because we want to be able to study how the neuronal basis of many different things, so behavior, perception, and everything. So to know how in the circuits at the neuronal network, at the neuronal scale, all these things are encoded. So this is why we are going to study, we use optical methods. And so now this is the overview of what I'm going to present you today. So first I'm going to do an introduction showing the optogenetic tools that we will use then in the techniques that I will present later, and also present the main challenges that that other people already introduced before, which are scattering and optical sectioning. Then I will go to a first class of techniques to present morphological imaging and then imaging of neuronal activity. So first, optogenetic tools. So we heard of them already several times during this course, and probably you're already familiar with them. Just a quick reminder. So the optogenetic tools, the general definition, read it, it's genetically addressable molecules that enable the use of light to monitor and to control biological function. So it's something very general. And the main principle is this. So you have a peptide or a protein that is encoded by a gene that you insert. So you insert the gene in the organism, then it expresses the proteins. And sometimes you need a cofactor that could be there or not be there. Sometimes you need to introduce it. Sometimes there is no cofactor needed. And then you have your optogenetic tool that is sensitive to light. And you can do different things with this optogenetic tool. So you have either reporter or actuator. So I'm not going to go too much into details, but reporter, they will change their light emission in response to parameters of interest. So for us, I will focus on two different parameters of interest. One in the concentration of calcium, And you will see in the next slide that this allows to look at activity. And one is directly the transmembrane voltage, which is the direct measure of the activity. And the other class of technique that I won't go too much into detail today because I'm really focusing on imaging, but it's very important, this class of techniques in neuroscience these days is actuators. And so, for example, the main actuator that has been used is some channels that can be opened through light, such as channelodopsin. And the idea is to then elicit activity in the neurons using light. Okay, so first, optogenetic reporters of neuronal activity. The main one that is used by the community is genetically encoded calcium indicators. And the most famous one is this GCAM here. It's based on a GFP protein, so a green fluorescent protein, to which several cofactors are attached. And so the idea is that when this protein is in presence of calcium, it will, the calcium will bind, and the protein conformation will change, and the fluorescence will change. So this is an example here in this paper where they presented GCAM6. So here there is an electrode that is inserted into one neuron, and they are looking at the fluorescence of the neuron with time. So they record both the electrical activity with the pipette here and the fluorescence. And you see that every time that a neuron an action potential is emitted, you have an increase of the fluorescence. So because there is calcium that is, the intracellular calcium inside the cell is increasing the level. So basically, if you measure this blue curve here, you might be able to guess what is the activity. So as I said, it's very widely used in the community. Most people use this technique to measure activity. And it's based on the best sensors are these GCAM sensors that are based on GFP. So they are green, and this is important for the future of the talk. And as I said, it gives an indirect measurement of neuronal activity, which means that it's quite challenging sometimes to really recover the activity from these traces. So one of the challenge is that in different neurons, you will not have the same signal that is corresponding to a single action potential. So then it's going to be difficult to know how many action potentials are emitted by which neuron and everything. So there is a huge variability. And also, if you look here at the response to a single action potential with different calcium dyes, so the GCAM8 is the last one that is quite fast, but even if it's quite fast, you can look at the red curve or the blue curve here, it still lasts for several hundred milliseconds, so maybe 100 milliseconds or a bit more. So, and you know that the action potential is 1 millisecond, so this is way longer than an action potential. So if you have fast bursts, for example, it might be difficult to find them with these techniques. But still, it's a very nice technique. And also another thing is that you have calcium signal in the brain that is not related to action potential. So maybe you can be confounded sometimes. But still, it's a very nice technique. It's very widely used. And the fact that it's slow, the response, it's actually an advantage because you have more time to collect photons. So this is quite nice in the end. And I mean, as a proxy for activity, it still gives nice results. And also, something that you can do is you can also use directly this calcium sensor to measure calcium signaling and not activity, which some people are actually doing. So now, if you want to measure directly the activity and not going through calcium, there are optogenetic reporters of voltage indicators. So they directly measure the membrane voltage. And so those are very new. So right now in the field, there is an explosion of all these techniques. So many labs are developing new voltage indicator. And it's becoming the thing to be very excited in the field. So here is an example from a few years ago. It's just an example of a voltage indicator, which is called ASAP3. It's made of a voltage-sensitive domain, so which is inserted in the membrane of the cell. And when there is an action potential, this voltage-sensitive domain will change its conformation, and it's attached to a circular GFP, which will change its fluorescence. So here is an example again where they put a pipette, electrophysiology pipette in the cell to measure the action potential, and also they measure the fluorescence at the same time. So you see the fluorescent curve here. So compared to the calcium signal that we had before, you see two things. So first, the signal is quite small, it's 10%, whereas before the signal could be maybe 100%, so much larger signal here. And also it's very short. It's a few milliseconds. So, but you see it's really well following the voltage. So it's a direct measurement of neuronal activity. And now there are many different tools that exist. I'm not going to present them, but there are like tens of tools that exist with all their pros and cons. And most of them, they have kinetics that are on the order of milliseconds, and the sensitivity they give in vivo, the delta F over F for one spike is usually on this range of 10%. But actually, it's a very big challenge to image these tools because the signal is very fast, so you don't have a lot of time to collect photons, and also it's very small, it's only 10%. So you will see later in the talk that it's quite a challenge to look at these voltage indicators, and people need to develop new techniques that are adapted to look at these voltage indicators. Okay, and just briefly, the other type of optogenetic tool that is widely used in neuroscience is this channel rhodopsin, which is a channel that opens with light. And you see that if you express a lot of channel in your membrane and send then blue light on the membrane of the cell, you can elicit action potential. So this came about 20 years ago, and it made a huge revolution in neuroscience to be able to control. Yeah.
Sorry, maybe I missed it. When you say it modifies the fluorescence, when it's the protein change, is it because it absorbs more of
This?
the light? Is it lifetime change, frequency change?
It's the fluorescence that change, the amount of fluorescence that is emitted that change. So when for example here, when there is no calcium bound to the protein, the GFP is in a state where it's almost dark. I mean, the fluorescence from one molecule is very small, but then when it binds protein, the environment of the GFP changes, and it becomes, the pH changes, and it becomes brighter. So the lifetime is the same, the decay time is the same, everything is the same, just the amplitude of the fluorescence is changing. Yeah. Okay. So for this channel, it's so then you send light to the channel, and it opens, and you can elicit action potential. So as I said, it was a huge revolution in neuroscience. And there are many different types of channel rhodopsin now, some that are very fast, some that are very slow, some that are redshifted, which is nice if you want to try to combine it with this GCamp indicator, even though this is quite a challenge. And some that instead of eliciting action potential and so depolarizing the cell, here they are hyperpolarizing the cell, and so it's the other way around. So I'm not going to go into details because I don't have time to discuss this today, but this is also something very important in neuroscience. So how do you get the expression of these optogenetic tools? Basically, there are two main techniques. One is to use transgenic mice lines, where actually already your tool has been genetically encoded inside the ADN of the mouse, and it's already expressed. So I have two examples here. And what people most commonly do is to actually inject the AAV that is coding for the sequence inside the brain of, for example, here a mouse. And so then you can inject your genetic tool exactly in the area where you need it to be. So you have an atlas of the mouse brain with all the small regions, so you know exactly where you need to inject. So then with the stereotactic arm, you point to the region where you want, and you inject your AAV. So then you can really be very specific of which region you're going to look, And also, because you are injected some AAV with a genetic sequence that has some promoters, you can also choose which kind of cells will be targeted and which. So, for example, if you're interested in specific type of interneurons, you can express your tool only in these neurons. Okay. So what is the advantage of this technique compared to electrophysiology, which is the gold standard for looking at studying the brain, or used to be the gold standard until a few years ago? So first, it allows access to many different cells at the same time. So this is an example where I think they have access to a bit less than 1,000 of cells at the same time with cellular precision, and you can very well see the activity of the neurons. Also, as I was just saying, because of genetic targeting, you can choose a subset of types of cells that will express your indicator. So you are sensitive to specific population, and you can dissect what is this population doing inside the whole brain easily, which you cannot do with electrodes. And also you can look at projections. So for example, you can inject your tool inside one brain region, and your tool will be expressed in all the cells that project to this region, for example. So this allows to also study where the cells are projecting and also target specific projecting cells. And one of the most important advantage compared to electrophysiology is that you see the cells and you can image the same cells over and over. For example, here until 36 days, and you find the same cells again, and you can look at how their coding is varying with time. With electrophysiology, this is for now impossible. They can look at many cells with arrays of electrodes, but it's very difficult to keep seeing the same cells because they don't know where exactly are the cells. They are just relying on a signature, a signature of the electrical signal. Okay, so we are happy we have these optogenetic tools. So now we need to use them to look at the brain, but we have to face some challenges. We already heard from them in the previous talk. The two main challenges for imaging the brain is scattering and optical sectioning, and the need of optical sectioning. So just a brief reminder, we saw this curve already yesterday, this figure. So what is scattering? Scattering is when you send photons inside the brain, inside the tissue that is with many different particles of different size. The small particles, they will diffract the light, so they will change the direction of the light. So in the beginning of the tissue, the light is not yet scattered, and you have ballistic light, so light that is not scattered, and then it will encounter some obstacle, and it will start to scatter. And so you see that we can define a mean free path, which is the distance between that the photon travel before encountering scatter, and it's very small. It's like 100 micron in the brain. So this is a very big challenge. So we can say that this scattering length, it's increasing a bit with the wavelengths of the light. So it's interesting to have techniques that are more in the infrared than in the visible for this. But it gives a very strong limitation, because if you look at because most of the optical technique, they work with ballistic light, or light that is not diffused so many times. So basically, you will use the first 100 to 300, 400 micron of the microns of the brain. But if you look at a mouse brain, for example, well, this is huge. This is 10 millimeters compared to this 100 microns. So basically, you have access only to the surface of the brain, and it's really a challenge to go deeper, and it's really needed because we are not only interested in the first 100 micron of the brain. So this is a big challenge, the biggest challenge. Then another challenge is this optical sectioning that Alessia already described. I just want to bring it back because it's so important, I think, this notion. So I just want to remind you the concept. So if you're imaging a large tissue, what you want, ideally, is to get the photons that are in focus, that are coming from the focal plane. And those are sharp. They make a sharp image. But in general, if you don't do anything special, you will get also many photons that are coming from the out-of-focus plane. And they will create this kind of image where you have an out-of-focus background that is blurred and that is hiding what you really want to see, which is this. And the physicist, the optician that try to quantify this, they use a specific way to quantify that I want to introduce because I will use it later. So what we do to quantify this is we use fluorescent planes. So it's just a fluorescent plane of, just a plane of fluorescent probe. It could be fluorescein or anything else. And what we do to characterize the optical sectioning is we quantify what is the signal that, with our microscope, depending on the distance between this fluorescent plane and the focal plane of the microscope. So this is what we have. We plot this curve, the signal, depending on the position. Okay. So what you want ideally, if you have an optical section technique, is you want a high signal when the fluorescent plane is in focus, and then you want the signal to decrease when it's out of focus. You don't want to collect this out of focus photon. So this is, for example, the green curve here. The green curve here has a maximum signal in focus and then decreases. But so this is a technique with optical sectioning, but some of the techniques, they don't have optical sectioning, and then you have exactly the same signal no matter where is your plane. And this is a problem, and this will lead to these images. Okay. So now I just want to come back to the most widely, the first techniques that were developed that were already introduced by Alessia just to understand if they provide optical sectioning and how are they robust to scattering. So just to make our ideas clear. So the easiest, the most simple technique that we can use in fluorescence imaging is conventional wide field microscopy. The idea is just you bring light, so usually a LED or a lamp, and you illuminate a wide field of view in your sample. Then you collect the light with your objective and you focus it on a camera. Okay, so very easy. You just need a dichroic mirror to separate the laser, the light, illumination light from the fluorescence. So, in your opinion, does this technique provide optical sectioning? Okay, I see a few no. So no, it doesn't provide optical sectioning. So actually, if you look at a movie that is that you can register from the brain, so the small points here, they are neurons that are active. You can see some activity, some neurons that are flashing. But you see that this activity is buried in a huge amount of blurred signal. And so it's very difficult, actually, with this technique to find the signal from the individual cells. And if we come back to our optical sectioning technique, the characterization, it gives a flat signal with the fluorescent plane as a function of Z. Okay. What about robustness to scattering? It's not great either, because when you have scattered photons here, scattered fluorescent photons, when they will reach the camera at a place that is not supposed to be the place where they should reach the camera, and you will get a blurred image. So scattering, as soon as you have diffuse scattered photons, they will blur your image. And so basically, you will be limited to a maximum depth of imaging that is about this scattering length, so 100 micron. So easy technique, but not so good in terms of sectioning and scattering. Okay, now confocal microscopy, that was described also by Alessia. Do you think it provides optical sectioning? Yes, it does. Okay, so let's just come back briefly to why. So imaging, you have a point source in your sample. Let's look at this point source here, which is out of focus. This out of focus point source, because you use focused, so in confocal imaging, basically you focus a laser beam inside the sample. So you focus your laser beam here at the focal plane, but your point source that is out of focus is not going to be that much illuminated because the light will have diverged, and so the intensity that reaches your point source is small. Also, so it will emit a smaller fluorescence than if it was in focus. And also, the fluorescence, it will be focused here. And in confocal microscopy, you have a pinhole in front of your detector. And you see that, well, it's not focused at the pinhole anymore because it's out of focus. And so most of the fluorescent light will be blocked by the pinhole. So you have two reasons why you have optical sectioning. You have because you focus the light, and because you use a pinhole. Okay, and so if you look at the curve, this is the green curve that I showed before. You have a very nice optical sectioning. And this image that I showed at the beginning, it's a rainbow image. It was acquired with confocal microscopy. So nice technique. So what about the robustness to scattering? Okay, so let's look at what happens in a tissue now. So in a tissue, you have your laser that you want to focus here at the focal point. But you see that some of the photons, they are scattered on the way. They are not reaching the place they should reach. And so if they are emitted photons, even if these photons are not scattered, these photons, they will not reach the pinhole. So that's good. We don't want to collect them because it's corresponding to a place where we are not trying to look at. We want to look at this place, and because of its scattering, the illumination went here to this place. So we are rejecting all the photons that are emitted from scattered in illumination light. And now you also have light that is reaching the place that you want to look at, which is the focal point of this objective here. And you see that some of the light is actually going to the pinhole. It's not being diffracted nor scattered on the way back, so that's good. And then you have some light that is emitted that is scattered on the way back, but again, it's not reaching the pinhole. It's blocked by the pinhole. So basically, by this way, you are selecting ballistic light, and so you are removing scattered light. So it's nice, it's what you want. But on the other hand, you can see that you will lose a lot of signal, because a lot of this illumination light will not reach your focal point, and because the lot of fluorescent photon will not reach the pinhole. So in a scattered tissue, in scattering tissue, well, you will lose rapidly some signal. So it's somewhat resistant to scattering, but not so much. So another technique that was introduced by Alessia, which I like, even though I never worked with this technique, I think the idea is really interesting, is to use light sheet microscopy. We heard of it yesterday also in the acoustic talk. Well, the idea is if you don't want to collect these autofocus photons, well, let's not image the autofocus plane. So let's illuminate only the focal plane. And for that, you use a laser that you focus, but on a perpendicular direction to the detection axis. So you illuminate a plane of light of your sample, and then you collect photons in the perpendicular direction, similar to with the wide field microscope. So I think it's a great idea. So of course it provides optical sectioning. It was built for it, basically. so this is great. What about the robustness to scattering? Well, it's not so great because you can see that the blue photons, they will be scattered for sure in a very scattering tissue. They will be scattered along the way, so it will enlarge the thickness of the optical sheet, so you will lose resolution, basically. And also, the photons can be scattered also at the detection, which means that they will not reach the pixel they were supposed to reach on the camera, and your image will be blurred. So it's not a very robust technique to scattering. It's a great technique for optical sectioning, but not so much for scattering. And something that we can say right away, it's the advantage of this technique, it is very fast, because you saw confocal imaging, you illuminate a point, and then you have to scan this point to make an image. Here, you can take the image right away. So if you illuminate the whole sheet, here the whole plane, you can take one image in one shot, and you get the whole field of view. So it's fast. Okay, last technique I want to introduce, because this is maybe the most important technique for imaging the brain. It's two-photon microscopy that was also introduced by Alessia. So two-photon imaging, as she said, what you do is you use two-photon absorption. So you have two photons that are absorbed at the same time, and then they will give rise to a fluorescent photon that the energy is more or less twice the excitation photons. Okay, so it's a bit the same as confocal imaging. So the idea is that you will focus a laser inside the sample, collect some light at each point of your laser spot, and construct an image like this. What is very nice about this technique is, first, it has intrinsic optical sectioning. And why is that? because you want to have this nonlinear effect. To get this nonlinear effect, you need to focalize the light and to have a lot of intensity. It's this two-photon fluorescence here is only coming if you have a very, very high intensity. It's nonlinear effect, so you need high intensity. It means that if you look at what happens when you focus your light, you will see a signal here from the focal spot where you have a high intensity, but outside of the focal spot where the wavelength, the light will have diverged, there is not enough light to create photons, so you won't have photons. So you have an intrinsic optical sectioning, which is actually exactly the same as the confocal microscope with this technique. Something that is very nice. Why this technique is very nice? It's because it's robust to scattering, and it's robust to scattering for different reasons. The first one is we use infrared light to illuminate. And we said earlier that scattering, it becomes the scattering free path. It increases with the wavelengths of the light. So the infrared light will be less scattered than the blue light that is used for confocal imaging. So this is one advantage. Another advantage is that the scattering excitation photon, even if they don't reach the focal plane, they will not create fluorescence because they will be too weak to create fluorescence. And last, and maybe the most important advantage, is because you know that the signal is coming from the focal plane, because this is the only place where light is emitted, then even if the photons are scattering on the way back, you can collect them, you know where they are from. So then the idea is to use the detection cone as large as possible to collect as much photon as possible and to put them in a detector. So you don't have any more a pinhole as in confocal microscopy. So for all of these reasons, two-photon microscopy is a better technique to look at tissue. It's more resistant to scattering, and it's been used in like most of the research using optical methods in neuroscience, I would say. Well, okay, just a quick summary of all this. Basically, we can just talk more about the two techniques so I'm comparing the 4 techniques that we have, depending on their robustness to scattering, optical sectioning, and speed. So I will discuss about the 2 techniques that have 2 green dots. Light sheet microscopy, which, as we said, provides optical sectioning and is fast, and two-photon microscopy that is robust to scattering and has optical sectioning. This technique is slow because you need to scan the point in the sample, so it takes a lot of time. Okay. Good, so I don't know where. Okay, time flies. I will be fast on this morphological imaging to have time for the imaging of light, activity. So morphological imaging, the first techniques that people use are not very fancy. What they had is a two-photon microscope, and they had a microtome, which is something to cut slices. And so basically the idea was that they were imaging the top of the brain, then slicing the brain, then imaging what is... So this is not for in vivo imaging. This is, sorry, this is for samples that have been, the brain has been extracted and it's been fixed with PFA and everything, and you just want to know the connection, what was fluorescent in your sample, or things like that. So you put your fixed brain underneath your microscope, image the first section, then peeled it, image the second section, peeled it, and by like this, collect the whole brain. So in this example here, which is quite old, it required 16 hours to image the whole brain, and also it creates a lot of data that is difficult to manage. So that was the first technique. And then I just want to say briefly, about at the same time, which was in the years 2010, there were an explosion of new techniques to allow to clear the brain. So again, it's mostly used for brains that are not in vivo. And so the idea is to remove the scatterers and to remove the pigments that create absorption and make the brain clear. So I'm not going to go into details to how this technique works, how they remove the scatterers, how they adapt the index of refraction to have the less scattering as possible, but you can see there are a lot of reviews on this. Just to show you 2 examples of the best clearing agent. One is iDisco, you see here, developed by Nicolas Régnier, and one is Clarity. And you see the brain, it has completely disappeared, so it's very efficient. And there are many alternatives, many different techniques, many different ways. It's a huge community that is working on these techniques. So how to look at this clarified brain? Well, you can guess that one nice technique for that is going to be the light sheet microscope, because it's fast and it has optical sectioning. Okay, fast optical sectioning, but now we want to look at brains that are very big. So the mouse brain, I remind you, it's 10 millimeters. So if you focalize your light sheet with a microscope, you see that actually you still gain this divergence of the beam. You still have a bit of divergence of the beam, even if the numerical aperture is low, so the angle of the beam is low, still it will diverge. And what you see on the border of the field of view is that the beam has diverged, and the resolution, the optical sectioning, they will be low. So there have been many trials to try to improve that, and I just want to briefly present one of them, which I think is very nice. The idea then was to scan this beam along this Y propagation here, so that the place where the beam is very small, it will move along this direction. And then on your camera, you use something that is called the light sheet mode, so it's named for a reason. And the idea is that you will collect light only in the pixels that will be corresponding to this scene sheet. And then with the rolling shutter of the camera, the pixels that are actually the lines that are on, that are actually looking at light, they will be swept. And so you synchronize the scanning of your sheet of light with the rolling shutter of the camera, and you get some very nice image. Just to show you here. With these techniques, you get very clear images of the brain, which, if you don't use them on the border of the field of view, it's blurred. Okay, and just an example of this. So here, it's an optical clearing technique where they use what is called U-disco. The technique is both clearing the brain and also shrinking the brain. So this is also a good advantage, because if you shrink the brain, it's easier to look at it with light sheet imaging. And just to show you the images that they get, you can, I don't know if you see very well, but here you see some dendrites. So you see dendrites here. So you image the whole brain and also the whole nervous system. And you can see dendrites. So you have all the scales, all the special scales that you want. It's quite amazing. And just one last word about this. Will this work in vivo? So recently, a few months ago, in September last year, there was this paper in Science, I think, that came viral, where actually what they did is they introduced a food color called tartrazine inside a living animal, a mouse here. And what they showed that because this tartrazine, if it's in solution, it increases the index of refraction of the solution. And so then the solution has the same index of refraction as the lipid droplets. So there is no scattering anymore, because everything has the same index of refraction, more or less. And so by doing so, they managed to clear this mouse in vivo. And so here they are looking at imaging of internal organs, and it works pretty well. So this might be a huge change in the field of imaging. This is to be continued. Okay. So now moving on to the last section, which is how to measure activity, which is really what I work on. So I said the challenge is the scattering. One of the challenges is the scattering. So, well, why not using a non-scattering animal? So this is what a lot of people are doing. They are using zebrafish larvae, as shown here, and they are doing, they can image the whole brain very fast with light sheet imaging. So this is quite nice, but the zebrafish larvae, the problem is that they are very young animals, so you cannot do this in adult zebrafish, only in the first stage of development, so you don't have access to many behaviors and everything. So I would just want to cite the work that Thomas did during his postdoc, where they actually introduced another fish that is called Danionella translucida, which is transparent also when it's adult. And so this might be a specimen that can be used also for to understand how the brain is coding some behaviors in the adult. Okay, but so as I said, so if you want to use to look at mice, which is a nice model for imaging, or rats or things like this, well then this is not transparent. For now, we have no clearing, in vivo clearing agents. So we need to use techniques that are robust to scattering. And the actually most widely used techniques is two-photon imaging. It's the gold standard for in vivo imaging. So I just want to say briefly what are the main performances of this technique. So the speed, usually it's about 10 hertz, but if you use resonant galvanometers, it can go maybe up to 40 hertz. The penetration depth, it's about 2 to 3 scattering lengths. I think it's about 500 micron. Field of view about 500 micron also, and lateral resolution smaller than a micron. And here I show images, typical images of what people can look. So you have movies here. So you see very well the cell bodies, you see very well the dendrites, and you see the signal-to-noise ratio is quite nice. So these two-photon imaging techniques, it's a gold standard. It works pretty well until only 500 microns, so it is not very deep. And also something that is quite fun, it can be coupled with other techniques. So for example, it can be coupled with virtual reality. So here the idea is the mouse is awake. It's walking on this ball here. And while it's behaving and walking on the ball, the researchers, they are projecting some virtual reality to the mouse. And the mouse is actually walking inside a maze, like a corridor. And while the mouse is walking inside this virtual corridor, with two-photon microscopy, they are collecting the activity of the neurons, and they can find what has been found by by electrophysiology. They find place cells, so they find cells that are active in different parts of the corridor. Okay, so this is also a huge field to develop these virtual reality systems to be combined with these two-photon head-mounted microscopes. Okay, so now starting from this gold standard or two-photon microscopy, can we do better? So I want to show that we can do better in 3 different directions. So to go deeper, to go faster, and to adapt it to free-moving mice. So let's start with going deeper. So one kind of brutal way to go deeper is actually to insert a lens inside the brain. And the lens that people use is those green lenses. So they are very thin lenses. I mean, thin. It's 500 microns, so for you it's thin, but compared to the brain of a mouse, it's not so thin. So here is an example here with one green lens. They imaged a region that is 5 millimeter deep inside the brain, but you see the size of the lens compared to the size of the brain. It's still quite invasive, I think. Okay, there is a huge field of research also that is trying to use thinner endomicroscopes, so thinner things that you put in the brain to look at to do imaging. And one of the ideas is to use multimode fibers. So they are very thin. It's 200 microns. So compared to these 500 microns, it's much thinner. It can be even 100 microns. But the thing is, it's difficult to, if you look at light that propagates inside these fibers, so if you focalize the light at the entrance of the fiber, what you get at the end of the fiber is a speckle. It's like a scattering media, basically. So how are you going to do imaging with this? This is a big challenge. And a part of the community, what they are doing is they are shaping the light that is coming inside the fiber so that the fiber is actually focalizing the light. And you can get nice spots at the end of the fiber, and then you can move the spot, create imaging and everything. So there is a huge part of the community that's working on this. There are reviews if you're interested. But for now, it's not working very well in mice, because the thing is, if you move this fiber, actually you need to adapt the electric field that you bring in the fiber in order to keep your spot, and it's very difficult, very challenging. So it's a big field, but still it's not, the work is not finished yet to make something that works very well. So let's look at another direction. Can we do non-invasive deep imaging? So what is limiting the depth into photon microscopy? Do you have any idea? Scattering, exactly. Scattering is limiting the depth, in particular because the pulse that you send, the illumination light, the intensity at the focal plane where you are going to look at, it's decreasing with the depth as this exponential law here. So when you go deep in tissue, you lose a lot of your excitation light because of scattering. So how to increase this signal? Well, one idea is to change the wavelengths, because we said that if you go to more infrared pulse, then it's gonna go deeper, and this has been shown by these guys here, where they looked at the vasculature with an infrared wavelength of almost 1300 nanometer compared to 800 nanometer, and you see that they can go much deeper. So it's great, it's a great technique. The only problem is that we have no calcium indicator that can be excited at this wavelength. Because I have said the best calcium indicator is this GCamp, and it's excited at 900 nanometer. So this is a nice idea, but for now it's not working well. So another idea would be to use more energetic pulses to increase the light of your laser. So this is what people are doing for sure. But at some point, if you just increase the intensity of your pulse, you increase the mean intensity of your pulse, and you are going to heat your sample. So you are putting too much photons, and the sample is going to heat, the temperature is increasing, and you're burning your tissue. So this is not what you want to do. But to keep the same idea, instead of just increasing the laser intensity, what you can do is you can change its repetition rate and have very intense pulse, but that are less frequent, and keeping the same average intensity. So let's look at an example to understand how it works. So here, between the two situations, I just multiply by 2 the intensity of the pulses, and have them spaced by twice the time that were spaced here. Okay, let's look at what we expect as a signal. So let's say that for one pulse of one excitation pulse, the fluorescence you will get is proportional to the intensity of the pulse to the square. This is 2-photon microscopy, it's nonlinear techniques, 2-photon absorption, so it's to the square here. Now, if we look at this interval of time here, where you have 2 pulses, well, you get twice this number of photons, so twice alpha IP squared. Now here, on the same time, you have only 1 pulse, but your pulse is excited with twice the energy. So here you have alpha 2p 2i to the square, so it's 4 alpha IP squared. So you have twice the intensity that you have here, the fluorescence photons. So it's a good alternative. I mean, it's a good idea to increase the intensity of the pulse and reduce the spacing between the pulse. Okay, so this generalized. You can, if you want to look at the math here, it's very easy, but I don't really have time to explain. So basically, you showed that the mean fluorescence it's proportional to the mean intensity of the laser to the square and the spacing between the two adjacent pulses. So if you go from TI-SIF oscillators, for which t is 10 nanoseconds, to regenerative amplifiers, for which the spacing between pulses is increased by a factor 10 to the 6, then you should go deeper. So actually it works. Oh, yes? Yeah, so then you will be still limited by the total intensity that you put, and also you will be limited by, if you keep increasing this intensity of the pulses, you will also create nonlinear damage in the sample. So you are limited by 2 things: the mean intensity, which is the heat that you deposit in the brain, and also the peak intensity of these pulses, which is creating some nonlinear effect that can completely damage the cells. So you need to find a range in between to work where you optimize basically those two things. And there is a review that I cite before. Later, you can have a look at this, which explains this. Okay, so it works better, but still it's limited in depth. You see that when you go very deep, your image becomes white. So why does it become white? Actually, it becomes white because there is a background that is becoming stronger than your signal. But so where is this background coming from? Because we said that light was coming only from the focal plane. So actually, the background is coming from the top of the slide. So let's just briefly look at why. So again, this blue curve, it's the sectioning curve. So now it's plugged in the lot scale, so that's why it doesn't look exactly the same as the one I showed before. But this is the signal coming from a focal plane, depending on the position of the focal plane. So it's maximum at the focal point, and it decreases out of focus. Okay, so this is without scattering. But if you add scattering, you have to add to this curve the fact that the intensity of the pulse is decreasing with Z. So it actually becomes this blue curve here. So it's a log scale, so there is a linear decreasing of this intensity here in the log scale. So you can see now, so this is the maximum of the pulse, but if you look, the signal that is coming from the top of the brain, it's actually not so little compared to this, and especially you will integrate a lot of signal here. So the signal will come from here, and you will have a lot of signal coming from the top of the brain that is uniform and that is that is higher than the signal you are looking for. So a nice idea that people had was to do 3-photon imaging, because now if you look at the optical sectioning curve of a 3-photon microscope, where you have 3 photons that are absorbed at the same time and not just 2, you get better optical sectioning. So if you look at the mass, the rejection of out-of-focus light, here it's in 1 over z squared, where z is the distance between the focal plane and the fluorescent plane. Now it's z to the 4th. So you get a much better rejection of out-of-focus photon. And now if you have this scattering, then still at the top of the slide, the intensity that is emitted is very low compared to the maximum intensity. So it's a nice idea. Also, if you go to 3-photon imaging, you will go to more non-infrared pulses. So this is also helping because the scattering length is going to be higher. So does it work? Well, yes, it works. This is a nice paper from the Chris lab. We developed this kind of technique. Here we can see in depth the comparison between 2-photon imaging and 3-photon imaging. So it's the same sample. They just alternate pulses for 2-photon imaging and pulses for 3-photon imaging to compare. And you see that the signal to background ratio is much larger here with 3-photon imaging. Same thing here with neurons. So you compare these two images that are acquired at 800 micrometer deep in the brain, and this gives a much higher signal to background. So it works well, and something that I like personally about these techniques is that they allow to look at the hippocampus, that is the region that is below the cortex, without removing the cortex. And so here is an example still from Chris lab, where they showed imaging of the activity with GCamp in the hippocampus. So without removing the cortex, because what I didn't show you before is with this technique that I showed, the 2-photon imaging, it was also, so sorry. It was this one. So this one, they were also looking at the hippocampus, but to look at the hippocampus, they had to remove the cortex. So they removed this, the cortex is 1 millimeter deep, they removed 1 millimeter of brain in order to look at what's below. So for sure, it's not that great. So I think 3-photon imaging can bring something to this imaging of the hippocampus or other brain regions that are a bit at the same depth. But as a word of caution, and also to answer to Alessia, you need to optimize your pulse. So you can have so if you increase too much the overall intensity of the laser, the average intensity of the laser, then you burn the sample, you put too much light and it's going to heat. And if you put pulses that are too high, you're going to create nonlinear damage. So basically there is a range where the technique works, but it's not a huge range and it has to be adapted. And for example, I think for photon imaging, I think maybe I mean, you cannot push this idea too much further, I think. But this is my idea of this. Okay, going faster. Oops. Until what time I have? So I will... 10 minutes. So I will go 2 minutes for going faster, and then 8 minutes for what I really do. Okay, going faster. Should I even go there? 10 minutes, you said? Okay, so I'm not going to describe how to go faster, just to say that it's a big part of the field right now to actually try to image this voltage die, and it's very challenging, as I said, because you both need to have, I mean, because the signal-to-noise ratio you need is very large. So I can explain that to whoever is interested later on, and the beautiful work that has been done on that. But I want to jump to the last session, which is the thing that I work on more. Even though I'm not going to present my work, I want to present this field because I also think it's very nice. Imaging in free behaving mice. So why would you want to do that? Well, so the technique I showed before, the two-photon imaging, it was on where the mouse was head fixed underneath the microscope. It's quite nice, but if you think about it, well, it's a bit stressful for the mouse because it has to be attached. And also, it limits the kind of behavior that you can do because the mouse is attached. So one part of the community is trying to develop techniques where the mouse is actually freely moving. So it's not attached to the microscope anymore. It can freely behave, which is less stressful for the animal, but also bring a wider range of behavior. Okay. But so what is the challenge for this? The challenge is that you need to have your microscope that is 2 grams. So because you need to put it on the head of the animal. So how can you miniaturize the technique and make them 2 grams? This is a big challenge. So the first method, of course, that has been miniaturized is wide field imaging. Because basically everything can be miniaturized in a wide field microscope. The camera is the same camera as you have on cell phones. There is a huge community that has tried to miniaturize these things, so we can use it now for fluorescence imaging, and so all the other parts of the microscope can be miniaturized. But as we said, this movie that I showed earlier was from one of these miniature microscopes. It's quite hard to have activity from single cells because of this out-of-focus background. Okay, so now, what about the two-photon microscope? Do you think it's possible to miniaturize a two-photon microscope? So this is a benchtop two-photon microscope from the Allen Institute. You see what it looks like. I put back here the schematic of the two-photon microscope. You have an infrared pulse laser that is scanned with a beam scanning unit, an objective, then lenses, and a detector. So do you think it can be miniaturized? What do you think can be miniaturized, not miniaturized in this? Not the source, for sure. You're right, not the source. The scanners. So the scanners, there was a huge piece of work on the scanners, and now they can be miniaturized. So the great thing is everything can be miniaturized except for the laser. But this was a lot of work. So for many, many years, for decades, people have tried to miniaturize the two-photon microscope and make images in free behaving mice, but it was not working. And then suddenly there is this group in China, the group of Liang Lichen. They got a huge grant, so like 2 million euros, I think. And they said, yeah, let's do it. But then they realized it was actually difficult. They didn't know when they applied for the grant. And then they went to see every single lab in the world that was working on this field to understand what was difficult, what was the challenging and everything. And they had a lot of money. So they also went to companies and everything. And so they made this. And this is the first miniature microscope that was working, two-photon miniature microscope that was working in free behaving mice. They use a hollow core fiber that is made especially for this project. So it's a fiber that will transport your 920 nanometer pulse without distorting them. because it's quite a big challenge to transport these infrared intense pearls inside fibers. They use a MEMS fiber. This is the thing to move the light. That was made especially for them also. That can rotate, so it rotates the beam very fast. Those lenses, they were commercial from a green tech company, and they also made on purpose this huge fiber bundle to collect as much light as possible. And they managed to get very good PSF, very good point spread function. So the point spread function is, the resolution is less than 1 micron. And yeah, and let's look at what they have. So, but also one thing that, yes? Sorry? A MEMS is so it's a micro electromechanical mirror. I mean, it's a micro mirror that mirror, yes, that can be controlled quite fast by. No, just one element. It's just one mirror, and that can be flipped very fast with an electrical signaling. Okay, so one thing that I really like on this paper, this is a great paper, by the way, if you want to read it, is they actually compare the signal that they have for a benchtop microscope, a huge microscope that you build on your optical table, and their tiny new microscope. And this is the comparison, so the benchtop and the miniature microscope, and you see that the signal is the same. So they use the same numerical aperture, of course, if you want to compare. But the signal is the same, the resolution is the same, and if you look at calcium imaging, the signal-to-noise ratio of the recording is the same. So it's really amazing. And then what they did is they did crazy behaviors to show that their microscope was working even with crazy behaviors. So here, the mouse is suspending by the tail. And you see image without and with motion correction. You see the dendrites very well that are turning on. Here the mouse is descending a platform, and here the mouse is having a social friend. But anyway, so it was kind of working, but there were limitations still. And in particular, the field of view was not so big, and things like that. So the guy that did his PhD in China, in his group here, then he went to Norway, to the Moses group, and he continued working on this project, and he built a new microscope that is here, that is very impressive. So it's the integration, the mechanical integration is very good. So they again use a MEMS scanner, again use same fiber to illuminate. But now what they have is they have here an electrically tunable lens to focalize the beam in the sample and to do 3D imaging. And they also optimize the fiber bundle to collect light to make it much thinner. And so by doing that, what they have is they show that the behavior of the mouse compared to a control with this microscope on the head is exactly the same as if it didn't have the microscope. And so here is a movie that they are showing on the paper. So you will see a mouse in 1 minute. Yeah. So you will see a mouse that is climbing this ladder here, and you see the image on the left, so without and with motion correction. And now the mouse is going to jump the ladder, and you can look at the image at the same time that the mouse is jumping. Hey, it's a bit scared, but he will do it. Yes. And you see nothing moved. So, I mean, the mechanical integration of this microscope is completely crazy. I think it's very huge work, and probably they have an army of engineers that work on this to make it very good. But what I like is that they are making it commercial and open source, so that people can actually buy this and be helped to use this in their lab. And so you see the microscope, it's very tiny. It's less than 2 grams, I forgot to say, but yes, it's less than 2 grams. It's 2 grams, I think. Okay, and the same group in China, they did 3-photon imaging with the same idea. So they have this microscope, and they also are able to image the hippocampus below the cortex with a 3-photon head-mounted microscope in free-living mice. For that, they had to optimize the microoptics to collect the the scattered light very efficiently, and the resolution that they have is very good. And so you see, they can make imaging of activity in the hippocampus below the cortex during 100 minutes, and the signal during 100 minutes is very stable. So I think it's also quite impressive. And just to finish what I'm working on in one slide, so I do confocal imaging, confocal imaging with this microscope here. And I also want to apply this to free-moving mice, but instead of miniaturizing the microscope, what we do in our team is we actually use those image guides. So they are basically arrays of fibers, and they use as a transmission of images between the microscope and the animal. And so basically, to do confocal imaging, we image with some lines that are scanning, and we transmit those lines inside the brain. Then we collect the fluorescence and bring them to a camera. And our detection lines are made on the camera with the same light sheet mode that we saw earlier for the light sheet. And so I don't know if the movie is going to work, but recently we are trying to look at voltage imaging, and this is the first imaging of voltage that we did a few weeks ago in free-behaving mice. So this is to be continued, but I want to say that this is also possible maybe to do this voltage imaging in free-behaving mice soon. Okay, so as a conclusion, I show you numerous methods, and they all complement each other. And there is a wide community that is pushing all the limits of the microscope further away. So we can go to a higher speed that I didn't have time to show you. I'm sorry. We can go to larger depths, application to free-behaving animals, which I showed at the end. Alessia discussed a bit about having smaller spatial resolution. There is a huge field of people that are working on this as well. And something I didn't really describe also is trying to have a larger field of view and have access to many, many neurons. So I just put one slide here, where what they did actually in the mouse is they replaced the skull by a crystal skull, so it's a glass skull. And so now they open a window that is covering the whole cortex. There is 1 million neurons below this crystal skull that can be accessible with 2-photon imaging. So that gives a lot of things to work on. But of course, as Alessia said, you need very expensive objectives because you want a wide field of view and a good resolution. So basically, there are a lot of techniques, and the idea is to find the best technique for your the application that you want. And I just want to say that all these optical developments, they are made in synergy with other domains. So we saw at the beginning the biochemistry domain to engineer the optogenetic probes. But there is a huge field which is data analysis that I didn't discuss at all. But of course, when you look at your image, you need to analyze them. And right now, we cannot go without very fancy algorithms to, for example, for imaging neuronal activity, to extract the neurons inside the image, to extract the activity, to clean the data and everything. So this is also a huge field of work. And with this, I'm going to finish. Thank you.